Have you ever stopped to think about how a brand new medicine actually gets the green light? It's definitely not a guess. It's this whole intensive journey, and the map, the GPS for that entire journey, is statistics. Today, we're going to follow that path, all the way from one single question to a discovery that could literally change lives.
You know, every big medical breakthrough really starts with a question just like this one. But when lives are on the line, a simple yep or nope just doesn't cut it. We have to know how sure we are. And that's exactly where statistics steps in. It's the language we use to measure our certainty and turn a mountain of data into a real decision.
So where does this whole quest for proof even begin? Well, it starts with a very formal process, kind of like a set of rules for a game, to make sure every single claim is tested in a way that's fair and totally unbiased. At the absolute heart of this quest is this powerful idea called hypothesis testing. Now, don't let the name fool you. It's not some dry, boring rulebook. It's much more like a scientific duel. We take our new, shiny idea and we pit it against the current champion, the old idea, in a really structured battle. And then we just let the data decide who wins.
Okay, so we've got the rules for our duel setup. Now we need a weapon. And in the world of statistics, our weapon of choice is data. So the very first step is to gather that evidence, carefully and with a whole lot of precision.
I mean, let's be real. We can't test a new drug on every single person on the planet, right? So we do the next best thing. We use something called sampling. By studying a really carefully chosen, representative group, we can make some surprisingly accurate conclusions about, well, pretty much everybody. Once we have all that data, we're not just going to stare at a boring spreadsheet. No way. We bring it to life with visuals. A plot like this one, for instance, gives us an instant picture of our results. We can see the mean, which is just the average, and we can see the standard deviation, which tells us how spread out the data is. It's really the first step in turning a bunch of raw numbers into actual insight. All right, the data is in. We understand its shape and what it's telling us. Now it's time for the main event, the hypothesis showdown. This is where we put our big idea on the line and see if the evidence actually backs it up. Now, to keep this whole process completely objective, we use this fantastic analogy. We put our idea on trial. And just like in a real courtroom, there's a core principle: innocent until proven guilty. So our starting point, our default assumption, is that the new drug does absolutely nothing. So in this scientific trial, we have two competing claims. First up is the null hypothesis. This is our innocent plea. It's the boring default idea that nothing is happening, no difference, no effect. Then we have the alternative hypothesis. This is the one we're actually excited about, the idea that our new drug does make a difference. Our whole mission is to see if we've got enough evidence to reject that innocent verdict. So, how do we reach a verdict? Well, it all comes down to one critical number, the p-value. The best way to think of the p-value as a surprisometer. It tells you just how surprising your results are if the drug actually did nothing. So a really, really small p-value means your results are super surprising, which should make you think, maybe my first assumption that the drug does nothing is actually wrong. So we need to get that p-value. But how do we do it? Well, statistics isn't just a single hammer. It's a whole toolbox, filled with different tools. And the tool we pick depends completely on the kind of question we're trying to answer. Let's start with a super common scenario you'd find in drug development. Let's say we have two different versions or formulations of the same drug, and we just want to know if they perform differently inside the body. Okay, here's our data. Just glancing at it, it kind of looks like formulation B has a higher average value. But, and this is the million-dollar question, is that a real meaningful difference, or is it just random chance based on the specific people we happen to test? To figure that out, we need a statistical test. So to answer that question, we use a tool called a two-sample test. It's designed specifically for comparing two groups. The software crunches all the numbers and boom, it gives us this output. The number to look for is the p-value, 0.0215. Remember our surprisometer? In science, there's usually a line in the sand at 0.05. Since our p-value is smaller than that, our results are officially surprising. This gives us enough evidence to reject the idea of no difference and conclude there really is a statistically significant difference between these two formulations. Okay, comparing two groups is one thing, but what happens when the journey gets a little more complex? What if we need to compare 3 or 4 or even more groups all at once? For example, imagine a company that needs to make sure the quality is the same across all of its factories. So here, we've got data on tablet weights from 3 different manufacturing facilities. We need to know, is the average weight the same for all of them? Or is one factory maybe producing tablets that are a little bit different?
For this job, our tool is something called analysis of variance, or just ANOVA. It's built to compare the averages of multiple groups. And just like before, we get a statistical output. The key result here is the p-value, which is 0.16. Now this time, our p-value is much larger than our 0.05 line in the sand. That means our results are not very surprising if you assume there's no difference. So our verdict is different. We failed to reject the null hypothesis. Now that doesn't prove the facilities are identical, but it means we don't have strong enough evidence to say that they're different.
And there you have it. We've literally journeyed from a simple question to gathering data, to putting it on trial, and finally to reaching a verdict. This whole structured process is how we go beyond just looking at things and into the world of real evidence-based discovery.
And this is exactly why all of this matters so much. This rigorous process gets rid of the guesswork and personal bias. It makes sure that the huge decisions made about the medicines we all depend on are based on objective, solid, statistical evidence.
I absolutely love this quote because it just nails the whole spirit of statistics. It's not about finding one single perfect magical number. That's often impossible. It's about using our data to find a range of believable truths. It lets us be approximately correct, which is so much more useful and honest than being precisely wrong.
This incredible journey from question to conclusion isn't just for medical researchers. It's a way of thinking that you can apply pretty much anywhere. So the next time you hear a claim, ask yourself, what's the hypothesis? And where's the evidence? You might be really surprised by what you discover.